Okay, we are live. Good afternoon, good morning, everyone, depending on where you are in the world. Welcome to our webinar today on driving microbiome science to improve animal nutrition and quality of life, hosted by DNA Genotek today. Thank you for joining us. If you can, please use the chat box. You should all be familiar with the webinar etiquette by now. Please say hello. Please let us know where you're signing in from. It's great to get a sense of the audience and where everyone is joining us from. So today's webinar is brought to you as part of the campaign for the Pet Care Innovation Summit, which is taking place on the 18th and 19th of May. And that's a business meeting bringing together innovators in pet health and pet nutrition to improve pet health and wellness. So if you're a business involved in that ecosystem, please do have a look at that meeting and join us if you feel it's appropriate and interesting to do so. I've got some excellent panelists for you today, and we've got quite an active session planned, including some poll questions and some Q&A sessions. So please do engage with that. Your participation is certainly welcomed and encouraged. So with that, I'd like to hand to Wendy Phillips from Diversigen, who's going to be hosting and taking you through this webinar today. Wendy, over to you. Thank you so much.
Thank you. Good morning, good afternoon, good evening for everyone. I am Wendy Phillips. I am a bioinformatician at Diversigen, which is a sister company to DNA Genotek. Today we also have with us Dr. Ryan Honecker, who is Director of Microbiology at NomNom, and Dr. Matt Panasevich. Matt, say your name?
You're good.
Panasevich. So who's as a R&D senior nutrition scientist at the Blue Buffalo Company. I'll be giving them a more detailed introduction shortly. First, as a reminder, this webinar is being recorded and you will be able to access it soon after the event. Today's discussion is inspired by the increased understanding of the connection between gut microbiomes and health outcomes. Better methods of profiling animal gut microbiomes have provided the tools to characterize the healthy animal microbiome and better understand the impact of microbial community on health outcomes. This in turn is leading to the development of better diets for companion animals that are more nutritionally sound and also help to prevent disease. Before we begin our discussion, we'd like to find out a little more about you who are joining us today. We are interested, and perhaps you are too, about the composition of our little ephemeral virtual community. To that end, we have just two poll questions. I'll bring up the first one now. We really appreciate your response to these. The first question is, how are you involved in animal nutrition? Is it with research, veterinary or clinical, a commercial application, or personal purposes only? I'll give everyone a little bit of time to familiarize yourself with the structure of the web app so that you can answer those questions. And then I'll be bringing up those answers shortly. Unfortunately, I cannot actually see how the responses, so I don't know exactly when to click. So I'm just waiting for the right amount of time, whatever seems like the amount of time. I hope that we will get some responses. So how are you involved in animal nutrition? Let's find out. Wow, that's a lot of people involved in research. That's great. A smaller percent in veterinary clinical and for personal purposes, and then another large portion in commercial application. We have one more question, which is, what stage of microbiome discovery are you involved in? Which sounds good for if we have a lot of researchers and commercial interests. So are you involved in early stage knowledge of the industry, microbiome study design, sample collection for analysis, sequencing and analyzing data. And some of these could be multiple choice answers. I'm actually not sure if you're allowed to select more than one. Again, I'll give you a little bit of time so we can see what everyone is involved in. Okay, 5, 4, 3, 2, 1, and the answers are, we have a lot of early stage knowledge of the industry, so it's super great that you're joining us for this webinar today. We hope that we can expand that knowledge for you quite a bit. A fifth of people doing microbiome study design, about a tenth of people doing sample collection, and almost a fifth with sequencing and analyzing data. So thank you so much for your participation. That's really good for us to know where the audience is at. So today's discussion is going to be split into 3 different themes. We're going to talk about first the link between pet health, diet, and gut microbiome. The next we'll do developments in animal nutrition and incorporation of microbiome. And thirdly, advancements in microbiome research and links to pet health. We'll spend about 15 minutes on each topic and then have about 10 minutes for questions at the end. However, you're welcome to post questions as we go, and I might be able to fit them in while we're still on that topic. Or I might wait to ask them during the Q&A at the end. Now let's meet our speakers more fully. Again, we have Dr. Ryan Honecker, who is Director of Microbiology at NomNom. NomNom is a companion animal health company. His work focuses on the intersection of the microbiome, nutrition, and personalization. NomNom has developed a direct-to-consumer microbiome testing kit as well as a robust online health assessment, providing digital reports for customers from both products. NomNom is utilizing these rich data sources to develop novel products and personalized experiences. Ryan holds a PhD from the University of Colorado at Denver and completed a postdoctoral fellowship at Stanford University. Thank you for joining us today, Ryan.
Thanks for having me. Thanks for the intro.
Yep. We have Dr. Matt Panasevich, who serves as R&D Senior Nutrition Scientist at the Blue Buffalo Company. In his current role, Matt is a diet formulator and investigates the application of novel nutritional technologies and their potential impacts on companion animal health. He received his bachelor's in animal science and a minor in nutritional sciences from Cornell University and has an MS and PhD in animal sciences and nutritional sciences from the University of Illinois, Champaign-Urbana. There, his research emphasis was on dietary fibers' effects on the gut microbiome and gastrointestinal health in companion animals. Thank you for joining us today, Matt. So let's dive right into this discussion. Again, please feel free to post questions at any time. And we'll go back to the topic slide so that you can keep track of where we are. Our first discussion theme is the link between pet health, diet, and gut microbiome. So let's begin with our current understanding of this topic. So my first question for our panelists is how do nutrition and the microbiome impact an animal's health? And what are the processes involved? I will let you two gentlemen decide who begins with how do nutrition and microbiome impact an animal's health?
Well, I think I can go first on this one. I think this is a great topic that we're talking about this, and I think really a lot of new innovation could come certainly out of what we know about the gut microbiome. And just thinking about typically when we thought about animal health and how we formulated diets, it was often to meet the nutritional requirements, of that of the animal, and based around standards built by the National Research Council. But now as we develop more ingredients, we know that consumers want nutrition above and beyond just the requirements. So things like around dietary fiber, we often would look at them as, do they provide good stool quality, good digestibility? And now we're starting to see that the links that in processes in the gut, like fermentation and short-chain fatty acids and their benefits to gut health, we see that the gut microbiome is playing a great role in this. So looking at new nutrition technologies that could influence these processes in the gut are really driving some new ingredients that we could look at to make our companion animal diets even better.
Yeah, I think it's one of the things that's really exciting about kind of this intersection of nutrition and the microbiome. I mean, there's always been in nutritional trials a huge amount of variability in response, and it turns out that one of the reasons is because there's a lot of variability in microbiome composition. And as Matt alluded, there are a lot of very specific things. Nutrition and diet intake in general is the major known driver of microbiome function and composition. And so marrying these two ideas together, introducing whether prebiotics or postbiotics or depending on what you're kind of looking at, introducing these specific parts of diet to have a positive impact that's rooted essentially in the microbiome itself, it's having a huge range of impact across a very wide variety of health indications.
Can you give us just a couple examples of those health indications?
Yeah, sure. I'm sure Matt and I can both, we can go off on this kind of indefinitely, really. I mean, there's not a day that goes by where there's not another study that's linking, correlating some sort of microbial composition with, any wide variety of health indications. Things that are really of interest to companion animal pet parents are, it makes sense that there's a lot of GI issues that are linked to the microbiome, because that's where the gut microbiome lives, is in the gut. So that one makes a lot of sense. But more nuanced things like behavior, anxiety problems, there are some really interesting links to the microbiome. Obesity is another one that's very, very complex, it's turning out, but it makes sense that there are some very strong links to gut microbiome. Things like allergies, so you kind of name it, across the board. There are correlations, and people really spending a lot of time and effort into figuring out how to prevent or ameliorate or mitigate symptoms across a wide variety of things, utilizing the microbiome and nutrition.
I think one good example to go off of that, we talk about the link with obesity and the microbiome. One of the flagship type of studies that were done and first high-profile cases in linking the microbiome to obesity was at the phylum level, where the Firmicutes to Bacteroides ratio is being linked to energy harvest. Well, this is based off of microbial composition. And over the past 10 to 20 years, I think we've really done an excellent job on the human side, and I would argue now we're getting more so on the companion animal side, where we're doing a good job at classifying what are normal microbiomes. And with changes in certain physiologic states, like with obesity, we could see some certain trends and certain changes in that composition. And now making that next step to linking those changes with differences in metabolism in the gut, fermentation, bile acid conjugation, neurotransmitter synthesis. How are these linking to host metabolism? And I really think that's the new and exciting frontier that we're finding ourselves in with the microbiome.
Yeah, and I mean the research is moving so quickly. It's interesting you mentioned the human space, because I mean human space is certainly far ahead of companion animal space. But I feel like we're making a lot of progress as well, and a lot of things are being translated from the human arena into dogs and cats, and as well into the agricultural space in general, like food production animals, for example. But things are moving. I mean, there's a lot of steam that's been picking up even over the last several years, and a lot of interest from bigger players in industry as well, starting to kind of see the writing on the wall for how important, what an important part of health in general this is. And then how it's also in, a lot of ways, the gut microbiome it's reasonably easy to get samples, to track it. The complexity is, that's one of the difficult parts is with every study, we realize there's a lot more things that we didn't know than we even thought that we didn't know before the study. But we're making a lot of progress. And what's really exciting, I think, is because of all this focus on in this research, we're learning a lot of really interesting things. We're starting to understand mechanistically why some of these things work. That we've known, fiber has been known to have positive health impacts for a long time. Well, now we're starting to understand why some of these prebiotic fibers and their fermentation byproducts and secondary metabolites, how these are having a direct impact, and mechanistically how these things are actually working. And with that increased understanding enables, better product developments, better understanding what's going on and how we can, again, develop products and treatments for pets and for humans as well.
Right, and one add on that, even within the technology of say fiber, we know that there's such a different array of soluble and insoluble fiber, and certain phytonutrients associated with different types of fibers that really, yes, the soluble fiber, the fermentability of that fiber is directly impacting the microbiome, but there's other components of these ingredients that might be having an impact on the microbiome that are really opening more of our understanding of these different ingredients.
Yeah, definitely. And then, not only that, but also, the matrix, which phytonutrients and which macronutrients and which probiotics or whatever are in which amounts, but then how those are processed also has a big impact. There's been some really interesting studies in companion animals and in people about, cooking and processing and all sorts of these things having, the microbiome, as I mentioned, responds to nutrition, to dietary intake, and, how food is cooked and processed and delivered has a huge impact on, what these nutrients are and end up being in the gut, and therefore how the gut ends up responding to those.
I'm going to just take a super basic question and one aspect of animal health, and it's not animal health per se, but maybe an indication of animal health, which is just say stool quality. Is there a way that you can that the microbiome might directly impact that or indirectly impact that?
Absolutely. I think one of the classic examples of something like this is one of the most common, when a pet parent and a veterinarian, one of the most common ailments are acute and chronic GI enteropathies, poor stool quality. And we know that through classifications of microbiomes, we know what is considered the major players in say, dysbiosis. And so really utilizing what we've known around microbiome characterization between a healthy and what would be considered as acute and chronic IBDs, we're able to actually pinpoint more so today than before that which ones could be actually playing, and how nutrition could actually influence those certain bacterial communities. So certain things around butyrate production around that is an energy source for the gut. We know that in many IBDs that seems to be less concentrations of that metabolite compared in healthy animals. So how we could deliver nutrition that could influence that, I think, is really been helped by what we know from our studies in the microbiome.
Yeah, I mean, stool quality is obviously something that is, that pet parents have a very direct interaction with on a very regular basis, right? But I mean, that's right. So much of the environment of the intestine is governed by the microbial composition that's there. So, how, intestinal permeability, these secondary metabolites that are anti-inflammatory, which kind of then help to set sort of this general baseline inflammatory level. All of these things have an impact on digestion and nutrient absorption and digestibility in general, which then have a very direct impact on the composition and quality of the stool itself. I mean, a huge percentage of stool in general is bacterial anyway. So these things are all very, very closely linked.
Thank you for those answers. So you're talking about the microbial community. How much do we know about what a healthy community looks like, or what an unhealthy community looks like? And are there major players, or where's our understanding of that right now in terms of what a good community looks like and what a not so good community looks like?
I think in terms of where the most development has been made on microbiome composition, really has been around that, what we've known of just characterizing what are the most predominant phyla. And it varies by what sequencing method you're using and at that point. But in general, we know that it's very similar in some of the major players that we see in humans. There's been a couple reviews on what a healthy microbiome is in dogs and cats, and mainly firmicutes, fusobacteria, Bacteroidetes, proteobacteria, actinobacteria. All of them are major phyla. Now, changes within that in terms of diet really do vary, but at least from the phylum level, from my reading and what I've seen, is that seems to be the normal microbial characterization.
It's a harder question than it sounds, right? I mean, in humans, people have been trying to define, because obviously you want to be able to define this is what's normal, and so any aberrations from this are abnormal or dysbiotic, and therefore you can start thinking about ways to track those and then return those to normal. But it's been, in humans, no one's been really able to figure it out. I mean, broadly, we know, kind of what's supposed to be there, but there's a huge amount of inter-individual variation compositionally that occurs, and which is really interesting to kind of think about in general, because these things are so pivotal to our health in general. But it turns out there's a lot of functional redundancy in taxonomy. So you can have two very disparate taxonomical looking microbiome profiles that actually have a pretty significant amount of functional overlap. And the function is actually what is largely more important for what's going on there. And so this is one of the things, that Matt alluded to, where as technology has advanced in sequencing technology, but also, proteomics and metatranscriptomics and metabolomics, all these different omics and then their integration is really helping us kind of better understand that it's more than just what's there, it's what it's actually doing. So, we do, Matt mentioned earlier, chronic enteropathies, there's been some great work done looking and understanding in some ways, things we know don't look normal, but as far as what things are considered really normal and healthy, that's been a lot more difficult question. There's something that we're obviously all of us are very, very interested in, so we can have a better understanding of it. But it's been a difficult question, but we're certainly getting closer all the time.
It's interesting on that. Well,
go ahead.
One more add on that, and Ryan brought up what's happening, normal, what's in the human side, and characterizing what's normal on the pet side. We think about our diet composition as humans, it could vary so differently, we're not having necessarily a complete and balanced diet every single meal of the day. One of the most critical things about being, on the pet food side is we are providing nutritionally adequate and balanced meals throughout the day. Any perturbations in that, there's different food forms, there's, that do have an impact on these communities. So we do see shifts in what's the predominant phyla. But we do know that, the varying diet composition seems more so on humans than what we see in our pets.
Yeah, it's definitely one of the advantages of being able to do this type of research in companion animals is that we have two things. One, we have ultimate control over what we're feeding, and we can report that in studies. And two, the dog's not going to lie about the pint of Haagen-Dazs that it ate, whereas in human studies, people, it's a little bit harder to get people to be 100% adherent to some of these regimens where you have a lot more control over your pets in a lot of ways. So yeah, we're hoping this is one of the things that if we can constrain this as a variable, then that it enables us to understand what that variable does when we alter it. So, giving a lot of power behind sort of, diet transition and supplement addition type, probiotic addition types of studies.
Before we move on, I was about ready to move on, but we do have a question from Noah about this composition, the compositionality of the community. And his question is, does the Firmicutes bacteroides ratio affect animal health? Is one phylum better than the other? And I'll tag on to that. Or does that depend on other factors? Like you can't just say one is better than the other. Or can you?
I think it really depends on other factors. You could look at within, say, Firmicutes, and there's certain fermentation or butyrate producing type of genera and families. You can find that in Bacteroidetes as well. To say that there is one bad phylum, I think would be misguided, and that's both, I think, on the pet and human side. I think overall, if you're looking at this from more of a 30,000 foot view, it's really around the diversity. That's been a lot of the linking how many different genera and phyla are present that really have been linked to just overall health. To pin it on whether one phylum is more than the other, it really does depend. It depends on multiple factors. Diet is very important in that, but it certainly could. It's not the only factor.
Yeah, I agree. I mean, these very broad, especially phylum-based, which is so high taxonomically. I mean, there's so much going on in all these. I mean, even strain to strain, substrain variation. Taxonomy in general is. In microbiology, as a lot of people probably know, there was just a big kind of reformation of the lactobacillus because things are just a mess because all this taxonomy is largely based on these, 100-year-old lab-based sort of assays. And then now in the genomic era, it doesn't really make a lot of sense anymore. So, these ratios, there's been, publications on, some of these ratios or like various ratios with Prevotella, for example, as well, but they don't always seem to hold up. And I think it's just because it's too broad of a swath at this point. And again, these finer taxonomic levels, and more importantly, again, sort of, these functional differences between these different organisms are kind of more important. Even things that are kind of traditionally associated with disease-causing organisms like Proteobacterium, there's lots of Proteobacteria in very, very healthy animals. And so what does that mean? Well, it just means that we don't really understand, and it's very hard to sort of make these very broad level assumptions. The more data we get and the more kind of metadata that we have associated with those, the better understanding we'll have, and we'll be able to start kind of picking apart some of these finer differences. But yeah, I think in general, it's really hard to use these very kind of broad ratios to be consistently meaningful. Diversity is a great example where there's been lots and lots of studies showing like in general, more diversity is generally better, right? But diversity is very kind of a broad thing. It means that there's more organisms there, which means there's more different functions and more sort of interaction, cross-feeding, all sorts of these complex ecological webs that are able to exist with increased diversity. But it's also not, there's plenty of lower diversity examples where things seem to be going just fine. So, these are the tools and the metrics that we currently have, but we're also all trying to build better tools and metrics as we understand that, the current ones that we've been using, kind of the best we had with technology. But again, as the technology and the amount of data that we collect gets better and better, our resolution and our metrics also increase in accuracy.
Oh, good. You're bringing us right to the next topic, which is developments in animal nutrition and the incorporation of microbiome in that. So my next question is, with sort of this all very complicated, complex community that you're working with, how do you create actionable data and then use that data to improve companion animal health?
I think for the first arm of that, of creating, the compositional changes that we see, how are we associating that with a clinical outcome, and or some systemic health benefit that we might be able to see? And I think that's really another leap. We're starting to go down that as the technology and the sequencing methodology increases. So, really, traditionally, when we looked at, say, a lot of 16S technologies, and we're characterizing the microbiome, we know the certain players that if we give a prebiotic, we know that this is a good bacteria. For instance, like a lot of the bifidobacterium or the lactobacillus. If we see an increase in that, that's generally recognized as a beneficial change in the bacteria. But now we know that there's many other players involved that are involved with butyrate production or other short-chain fatty acids, or have been linked to a certain, maybe they're lower in a certain disease state, and yet you changed it through dietary means in this way. Trying to link that back to a clinical outcome, I think, is really how we could try to link what we're seeing from microbiome to what the consumer and what they could get out of this improved diet or dietary technology that you want to apply.
There's a question from, and I'm sorry to interrupt you, Ryan, but this question ties right into what he was saying, from Adam, which is, how are you measuring outcomes for nutritional studies?
Well, beyond just even the microbiome, and I mean, we certainly do metabolites, not necessarily a full-blown metabolomics, and certainly stool markers of GI outcomes with like digestibility, stool quality, immunoglobulins, gut immunoglobulins are some examples, or even some linking that, and even just trying to see, do we get changes in serum biomarkers? And do we changes between serum and fecal biomarkers? What are their relationships? So those are some examples.
Yeah, I mean, the more metadata you have, the better the microbiome and more useful the microbiome data becomes, right? And so, if you do some of these multi-omic integrations, like Matt was suggesting, if you have clinical blood markers or metabolomic profiles from serum or from stool, even the more of those that you have, then you can start to really then make more direct correlations with the microbiome based shifts that you're observing. And then, this also goes back to just good study design. Once you start to see some correlations, then you start designing good studies to test these particular interventions, and look at what impact on, whatever the clinical metric of, clinical outcome or clinical biomarker or whatever it is that you're interested in, designing studies to test these exact things. I mean, we have a ton of correlational research in the microbiome space, but now what we're really seeing is kind of the second wave of, well, now we see all these things are correlated. What does that mean? Like, how can you intervene? What does this mean to change this dietary fiber? Like, what kind of impact is that going to have on real clinically meaningful outcomes? Part of, I think, what's, and it's going to go back to the first question, advances in the technology, and this is anything from sequencing technology, moving away from 16S into, more shotgun-based approaches, as well as integration in different types of omics, and even, the analytical pipelines for this have changed and gotten so much better. The statistical approaches have developed kind of in stride with these advancements and the size of these data sets. So everything is kind of moving together to make this better and better. But to me, it goes back to, the more data, the better the resolution in the microbiome data and the clinical biomarker data, as well as the metadata, that's really where sort of things start to take a little bit of shape, and then kind of making an iterative process where you go back and then test specific hypotheses and observations and correlations that you get from those first studies and from the existing literature that's already out there, to then build studies to specifically test these things. One of the things, really interesting things that is happening is that, sort of this personalization approach where we now know from a lot of human studies, and we see this in our own data as well, the baseline microbiome composition has a huge impact on what these clinical outcomes look like. So, you're starting to see a lot of this work in, there's been a lot of stuff with kind of IBD, IBS world in humans, as well as personalized cancer treatments. The microbiome role in that is having a huge, huge role. And it turns out that, a lot of it depends, your response depends on what you have there in the first place. There's just this crazy study with melanoma treatment where they changed people's microbiomes and they were not responding to chemotherapy. And just by changing the microbiome, a bunch of the patients became responsive again. And so understanding sort of what's there and what that not only predisposes you to, but how you will react to an intervention, be it nutritional or pharmaceutical or nutraceutical or combinatorial, that is kind of the next step. Obviously, that's incredibly complicated, but that's what this technology and the data set, the sizes of the data sets are starting to allow us to see into a little bit.
You are playing right into the audience's questions here. You touched on this, but I'll see if you can answer it a little bit more specifically. A question from Stoffel is the change in microbiome causing a disease, or is the disease causing the change in the microbiome?
Yeah, that's the question neither of us want to answer, but that everybody fights about all the time, right? Or is it a combination of both, right? So, the ultimate, it really depends on the indication, and ultimately we don't really understand it a lot of the time. So, I mean, there are lots of examples where, regardless of whether, a change in microbiome is causing disease or whether it's a byproduct of underlying disease, helping to rectify and correct and improve the microbiome can have a major impact on disease progression. So, in a way, that's kind of a cop-out answer. But obviously, this is a super important and very pressing question that everyone is really trying to understand. I mean, if I had to guess, I would guess a lot of the time it's probably a combination of both, where it's a feedback cycle where there's some underlying, there are certainly things, that are genetically based that will cause immunological defects or things that will cause a direct impact on the microbiome. But as far as kind of other diseases that are not genetically based, it's probably largely going to be compositional, or it's, lifestyle impacting, microbiome, which is causing disease, or lifestyle causing some disease, and the microbiome is responding to the environment, which then is exacerbating, the constellation of symptoms. It's all very integrated, and there's certainly, to my mind, not a straightforward answer to largely, almost any disease, even things like infectious disease. Pathogenesis has changed a lot from, Koch's postulates are not as straightforward as they used to be now that we're seeing sequences. Well, it's not just that you have, this E. coli that's toxigenic, it's that you also have these other populations which are pro-inflammatory. And so it's becoming more and more complicated, even for sort of what we're traditionally sort of thought of as simple, sort of straightforward microbiological cause and effect. Nothing seems to be that simple anymore.
And I would second, what I was going to say is, and Ryan alluded to this, ratio where this is coming from, I think the term is dependent on the disease or the ailment. When it comes to things that are impacted by lifestyle, I think that what the host can do, and whether it be dietary, exercise regimens that have been shown to have certain functional or compositional improvements to the microbiome, do shift the microbiome towards a more, say, a lean profile. And we do see a change in the phenotype, and we do, but other diseases, I don't think it's as clear cut. So it's another cop-out answer, I guess, too. But yeah, I'm in agreement with it. Just is dependent on what that disease could be.
It's really interesting too, because it's pretty hard to change a microbiome. I mean, a lot of studies show that, after an intervention ends, things most of the time will kind of go back to the steady state that they were at before. So kind of, thinking more broadly about what a shift means, like how do you redesign, and this is what, a lot of research is now looking at, is kind of these defined consortia where it's a bunch of bacteria, a bunch of probiotics, prebiotics that sort of interact in a stable way that then can try and shift the population in a more sort of stable way rather than just these transitory shifts. Although, although these can still have an impact on, ameliorating symptoms of disease, even if they're just transitory.
Again, you're playing into the audience's questions. Given that, you're saying that it's transitory. The question from John is, how critical do you consider it is to try and influence the establishment of the microbiome in the young animal instead of trying to, say, change it during the adult life? And I'm going to add on to that question. I'd like us also to get into what goes into the creating the food. So if you could talk about trying to influence, and is it important to do it in the young animal? And then both of you are in animal nutrition. How do you go about doing that?
Well, let me just say on this is, I think people are very used to what we know, what is in the literature on the human side, where the development of the microbiome after birth, and in terms of the companion animal side, the characterization of longitudinal change in the microbiome is still another area that maybe we're not as we don't have as much knowledge in. So a lot of the times when even developing a puppy versus an adult versus a senior-based diet, there are certainly different nutrient requirements and nutrients that we're going to use to optimize overall health. But as that being microbiome driven, I don't think we're necessarily there yet. But I mean, there's certainly things we could draw from what we've known on the human side that we believe will have benefit from development from a puppy through adulthood.
Yeah, it's a super interesting question for sure. I mean, and Matt's right. For companion animals, we don't understand it very well. We're starting to understand it a little bit in human space. There's been a couple really interesting studies looking at early life prebiotic, probiotic supplementation and long-term longitudinal impact on, especially things, autoimmune disease types of things. I mean, it makes sense. I mean, in humans, we understand it somewhat. There are these kind of discrete stages of colonization and microbial shifts, and then things sort of mature and are relatively stable. There's been a little bit of work done in pets, but not that much. I mean, I would certainly anticipate very similar things are occurring. And I would definitely anticipate that these early stage development and sort of is going to be very, very important longer term. But as Matt said, we don't have great data on that yet. It's something that, we're personally very interested in as well. I mean, if you can set the stage and have lifelong impact on, morbidity and ultimately mortality, of course, that's something that we want to try and figure out. But we don't have the data yet, but I'm sure it's going to end up being pretty important.
I think I might shift us into the next topic, which is advancements in microbiome research and links to pet health. And I was just thinking about a couple things that you said in particular, Ryan, about the interplay with genetic diseases. Do you see, or Matt, animal health using microbiome-driven precision-based targets? So, for example, maybe you could test a dog for a genetic disease and somehow also know sort of the microbiome that may benefit a dog with that disease, or sort of other sorts of precision-based targets with the microbiome.
Yeah, I mean, I definitely think this is where, in the human and pet space is moving, is using this as a tool. I mean, we kind of alluded to this at the very beginning, right? Where there's a wide variety of response to various interventions, and some of that is genetic, and some of that is, epigenetic, and some of it is microbiomic for sure. And so understanding all of these things together is going to be where we get the most power. I mean, the strength of microbiome-based interventions is that microbiome can be changed, right? I mean, the genetic backbone, understanding that is, of course, extremely important, and will shape the microbiome, and will shape, obviously, understanding of risk factors and so forth. Obviously in companion animals, we think a lot about, like breeds, and certain breeds have certain crazy problems because of like these, very weird inbreeding experiments we've been running on them for such a long time. But, thinking about, microbiome profiling as kind of a companion diagnostic where, you profile and understand the way you kind of get a blood test, and you look for changes over time. And are these normal for someone of your age and genetic background or lifestyle, diet choices? And when they change, why do they change? Is there anything we can do about them? But also using that as a way to understand, well, what nutritionally is going to be, there's a bunch of people doing this in the human space, these sort of personalized diets based on your genetics and your microbiome composition. Like, is the science there yet for that to be super accurate? Like, I don't know if it is, but it's getting there. And the more data we have, the more, genetic data we have, and there are companies doing great things in the companion animal space. And I saw one of the questions from Embark. They're doing some really interesting, great work on profiling that. There's a cat company called BasePaws. There's Wisdom Panel that's run by Mars. There's lots of genetic stuff happening. And all of this will kind of play into this understanding and personalization of approach. But thinking about, specifically microbiome is understanding what's there, and are you more likely or less likely to respond, or which, treatment for whatever drug, whatever condition you're interested in, which of these three treatments are you going to respond the best to based on your microbiome profile? Or do you need to take kind of a prebiotic, probiotic supplement while you're taking this medication to shift your microbiome in a way that is more favorable for this medication to be effective? That's where things, there's strong movement towards that. And I think we're getting there. It's obviously an extremely complicated problem, and there's lots of work left to be done, but we're starting to see some of those things sort of be realized, specifically more in the human arena, because, there's more data there right now. But that's kind of that's the most exciting thing to me, is kind of bringing all this back together and understanding, not only like the pharmacogenomics of this drug, but the microbiomics of how that drug is going to work on you, or on your pet at a specific point. That is kind of where the future of all this is heading, I think.
Matt, where do you see the future?
Well, I think Ryan gave a good comprehensive answer in terms of, a lot of these disease states and how a certain animal will respond based on certain outcomes. Where I think is really interesting, and a paper that's actually been referenced to a couple of conferences I've been at on the human side, the ZV paper in 2015 at a Cell, looking at integrating microbiome approaches with people's different responses, glycemic responses to certain foods. We know that they were able to predict, well, certain people, their response to certain foods, their glycemic index, their curve was different. And when they brought in the microbiome, they saw that certain microbiome profiles seemed to be, might be influencing this. And really trying to bring these together, and, even not just the pet that, is trying to respond to IBD treatment pharmacologically or nutraceutically, but what about our overall pet? What is the best diet that's going to, promote optimal health, and how do we define that? I think that is something that is of keen interest in the pet food industry. I also think too, in terms of from a diet formulator, and looking for new technologies, how we define some of these terms that are coming out. And what was brought up a little bit before on the topic of postbiotics, we've known a good amount about prebiotics, probiotics, and now ISAP actually just came out with a publication yesterday providing more definition around how we define what postbiotics are. I encourage people to, if they find that publication, to read that. And it's really interesting of, how it's looking at microbial products, non-viral bacterial products that could confer a health benefit to the host. And this is encompassing to companion animals, human, and livestock species. So I really think that, and these types of ingredients, whether it be probiotics, even prebiotics, postbiotics, they're added at very finite concentrations in diets. So as alluded before, diet is really, it's very hard to see dramatic changes in the gut microbiome. We only see that really in humans when there's like vegetarian or an all carnivore type of diet. We see these vast changes. But when we're adding something that's, a low percentage of the diet, which we often do on the companion animal side, understanding the functionality of that, of what that's doing to the microbiome is really crucial. And I really think a next step that we could venture into in qualifying ingredients to make our pets' diets better.
Thank you for that answer. I am going to invite all the participants to please send in your questions if you have any before we have to end the webinar, but I'll keep asking questions while we wait for anyone else to think of some. So we've been talking about animals in general, maybe said dog a few times. Is there a big difference between the needs of a dog and the needs of a cat, or the in terms of microbiome composition?
I mean, there's certainly a big difference in the amount we understand between between the two. There's a lot more work on, I kind of think of it as, the human space has the most, and then maybe dog is about 1/10 of the understanding, but cats are probably about 1/10 of the understanding of that. There's just not a lot known in cats. I mean, diet-wise, obviously, Matt can talk about the pretty fundamental differences in nutritional requirements, but microbiome-wise, they're pretty different. There's actually a lot of really interesting overlap between dogs and humans. There's been a couple of really interesting papers on this, which makes sense in a lot of ways, because until, the last 75 years or so, they ate the same food that we did, right? I mean, dog food as a separate thing is more of a recent invention. And so between sharing diet and environment, it makes sense that our microbiomes co-evolved in a lot of ways with dogs. But cats are obviously very different and, are borderline domesticated, I guess you could say even so. But, with that being said, that just means there's a lot more interesting things for us to uncover about them.
I like to call them the forgotten child, because it's exactly Ryan's point on we have a lot of fundamental research even around things like prebiotics that are very effective and have a lot of literature around their effectiveness in dogs, but not necessarily to the same breadth that we see in cats. And oftentimes it's assumed that the benefit is going to be the same. Now, we know that cats do have they are able to ferment carbohydrates in the hind gut. They do have that potential, so they can respond well to ingredients like soluble fiber. But understanding the differences in how much we feed of that and what's an optimal dose, and maybe their impacts on what that could do to the microbiome in comparison to dogs, I think would be something that we could definitely learn more on. Maybe making the assumption that the dog and the cat are the same, or we often even take technologies on the human side and based off of that literature, hey, we think this is going to work in the dog and cat, but has that been fully elucidated? Not necessarily with a lot of these technologies. So definitely opens up a lot of opportunities.
We have an audience question from Andrea which is, can you see a benefit of evaluating the microbiome in other companion animals like lizards and birds? Now I can imagine our understanding of lizard microbiomes must be way under that of cats, but I don't know, maybe there is a lot of lizard microbiome research.
Yeah, I mean, I think it's going to be as important in all animals as it is in humans, right? I mean, I think there's been a lot less work done on other companion animals because there's less of a market, there's less incentive for, companies to invest in their understanding. But I absolutely think that it is going to be important. I mean, I know there is work going on in birds. There's some work that's definitely going on in horses that I'm aware of. I don't know that much about lizards. I haven't really seen that many papers about it, but I haven't really been watching necessarily for them either. But I'm sure it will be, equally important in all species. And, as the technology and all these pipelines develop, it will become easier to apply it to other species. Obviously, like Matt was saying, massive differences between dogs and cats. There's differences are going to be even bigger between, cats and lizards or birds but the tools that we're developing now and the methodologies and the approaches will be applicable to those as well. And as the technologies get cheaper, it will become, easier and easier for those kind of areas to be explored.
I have another question that has come in from Michael, which is, how does the quality of ingredients impact gut health? He says MDM protein versus whole muscle protein.
So I believe that's a frontier and a question I think that's been posed on the pet food side. It's often, the pet food industry is very ingredient driven. I do think there will be more questions around that of what, certain quality of ingredients impact on the gut microbiome and their implications to certain health outcomes. He did give an example there of protein, and not sure on the differences for that per se. But one I would like kind of just, it spurred something in terms of protein source. We talked a lot about fiber and everything, but the development of these also like plant-based proteins that, from a formulation standpoint, bring low amounts of ash and minerals and make them very favorable towards formulating, are also, I guess, high in protein, or good protein sources, but could bring other aspects in of other metabolites that could be beneficial to the gut microbiome. So looking at different protein sources and their impacts on the microbiome, or potentially how they're processing, what processing aids and what's in their process flow that might be impacting health and the microbiome, I think is definitely another frontier for discovery.
Do we have time for one last question from the audience? We'll have a quick answer to Sue's question is it important to give dogs canine-specific probiotics, or can they receive the same benefits from human-specific probiotics?
Yeah, that's a very interesting question. There's not a lot of literature about that actually out yet. In the probiotics that we have formulated, we have found as many, host-specific strains as we can, because, ecologically it would make sense, and, that even if it's the same species, some lactobacillus species, bifidobacterium, whatever, even if it's the same species, it's going to be differentially adapted to live in a dog or a cat versus a human, right? So it makes some intuitive sense, some ecological sense, that it would do better in the environment that it has evolved to be in. But there's not great data really to compare the two. And also it depends, one lactobacillus, strains can be dramatically different for the same species. And so are those differences between two human derived or things that have not even been sequenced but have been approved? Like, are those differences as important or more important? It's kind of tough to say. That's kind of where we are right now. And, as sequencing and whole genome sequencing and characterization of, and understanding the function of how these strains work evolves, I think we'll be able to better understand that question. But it's kind of, I think it makes sense that they're probably more effective, but it's tough to say at this point. Super interesting question, though.
I think that kind of sums up the whole thing. We know a lot, but there's also a whole lot yet for us to find out. I think that's the end of our time, and I really appreciate, Ryan and Matt, you taking the time to give us some of your wisdom today and your the knowledge from your vast experience in the companion animal nutritional health space. And with that, I think we will be closing the webinar.
Yeah, absolutely. And thank you. So just to echo your comments, thank you, Wendy, for your excellent moderation and host of this panel. And Ryan and Matt, thank you so much for your contribution. Thank you to everyone for your questions and for engaging with the session. Really appreciate that. And just again, to reiterate, if you are interested in joining us for the Pet Care Innovation Summit that is taking place on the 18th and 19th of May. So we'd be very happy to send you further details on that. So wishing you a very good rest of your day, wishing you all the best, and thank you again to our excellent panelists. See you all later.
Thank you.
Thank you so much.
Bye. Thank you, guys. Thanks, everyone.
Bye now.